Okay, we'll get going. So redox biology now, we're halfway through the course. And today our first lecturer is Perwez Hussain. He got his PhD in India, then he did a postdoctoral fellowship in Switzerland, and we were fortunate enough to recruit him here to the National Cancer Institute. He's head of the Pancreatic Cancer Unit, and he leads the Pancreatic Cancer Special Interest Group in the Center for Cancer Research. His title today, Inflammation and Cancer, an Ancient Link with Modern Evidence. Perwez.
Thank you, Terry, for such a generous introduction. And what I'm going to do today is to discuss with you the evidence, both epidemiological and molecular evidence, that supports the role of inflammation in cancer. And I will highlight the role of inflammatory mediators, their complex biological interaction with carcinogenesis, with a special reference to chronic inflammatory diseases, and also some of the cancers, for example, pancreatic cancer. So let's start with this slide. And I just gave a search in the PubMed for the literature associated with inflammation and cancer. And in the last 10 years, there has been 28,970 articles, which shows that how widely it is studied and how vast the field is and how vast and complex the association is between inflammation and cancer. So the concept of inflammation and cancer is not new. About more than a century ago, Virchow put forward this hypothesis when he noted leukocytes in neoplastic tissues, and he made a connection between inflammation and cancer. And the recent evidence supports this hypothesis, are consistent with this hypothesis, and the link between inflammation and cancer may have potential implications in prevention and treatment. So this slide simply shows that the use of aspirin reduced the risk of cancer deaths. This is a collective analysis of 7 different studies from different centers, and they pulled together the data, and it involved about 23,000 subjects, which showed that the risk for several different cancers, colon, lung, and other cancers, the risk was reduced. So this slide shows an association between chronic inflammation and cancer. In cancer-prone chronic inflammatory diseases that can be either inherited, for example, hemochromatosis, hereditary pancreatitis, ulcerative colitis, and Crohn's disease, or can be acquired through a biological, chemical, or physical means through viral infection, bacterial and parasitic infections, and also metabolic syndrome, for example, obesity. And as you can see, about 18% of the human cancer, that is 1.6 million per year, are related to infection according to the World Cancer Report. So what happens during inflammation? And I'm going to give you an oversimplified presentation of what happens during inflammation and immune response and how it can be pro-tumorigenic. And it's a highly complex, but this is the oversimplified presentation. So a stress caused due to injury by physical, biological, or chemical means can launch immune response, innate immune response, and then adaptive immune response that can lead to inflammation, which generates a number of different cytokines, chemokines, free radicals, prostaglandin, growth factors, and proteinases. And that can interact with both epithelial cells and also the stromal cells and plays an important role in stromal to tumor interaction. This can lead to DNA damage, protein modifications, proliferation, and also microRNA regulations, a DNA histone methylation, angiogenesis, to mention a few, that can provide a pro-tumorigenic environment. In addition, stroma can also enhance metastasis through landscape effect. It's interesting that adaptive immune response through humoral immune response can enhance chronic inflammation, and it can also enhance Treg cells activation that can in fact help in cancer development and progression, but at the same time, it can also inhibit tumorigenesis through the cytotoxic T cell activity and also tumor cell lysis. So one of the important events during inflammation is the generation of reactive oxygen and nitrogen species. And also these species interact and give rise to reactive nitrogen oxide species. And these reactive species can induce a number of DNA damage, including point mutations and also post-translational modifications of important proteins that are responsible for maintaining cellular homeostasis. And this can happen either directly or indirectly through lipid peroxidation and generation of reactive aldehydes, for example, malondialdehyde and 4-hydroxynonenal. Inflammation can affect initiation, promotion, and also metastasis. During initiation, the reactive species and cytokines that are produced by immune cells can induce epigenetic alterations and mutations. Then it can transform the pre-neoplastic cells to neoplastic cells, and that can also start producing chemokines and cytokines, and they develop a feedback regulatory loop that can lead to survival, growth, advantage, and angiogenesis, and can also help in metastasis through enhancing EMT. And there are several cytokines and inflammatory mediators you will see later that plays an important role in the epithelial to mesenchymal transition, and that can help in invasion and motility and eventually metastasis. So there are several types of inflammation in tumorigenesis, as is outlined here. It can be, chronic inflammation, infection, and autoimmunity that can lead to mutation, genomic instability, tumor progression. It can be tumor-associated inflammation that can lead to tumor growth, survival, angiogenesis, and so forth. And it can also be caused by environmental and dietary exposure that can lead to mutation, genomic instability, and tumor progression, and can help in tumor development and progression. Obesity, which is a metabolic syndrome, is also considered as a chronic inflammatory disease because we see the generation of inflammatory mediators, including cytokines and also reactive species that can induce inflammation. And this can increase the cancer risk and progression. And what has been shown that the functions of adipocytes and macrophages during obesity shows a lot of similarity. In fact, there is a massive infiltration of macrophages in the adipocytes, and they both store lipid and also secrete several different cytokines, which is normally a function of macrophages. There are evidence both from our lab and also other investigators. They have shown that chronic inflammation can affect multiple pathways that are involved in tumorigenesis. This is just some example that free radicals and cytokines that are generated during chronic inflammation can lead to DNA damage and can induce P53 pathway, can inhibit RB pathway, can induce NF-kappa B signaling pathway, can lead to cell survival, but also at the same time, they can, and I will discuss it more later in details, that it can induce P53 suppressive pathway by inducing cell cycle arrest, DNA repair, and apoptosis. So the one of the inflammatory mediators that I'm going to discuss more in detail today is nitric oxide. And nitric oxide is generated during the conversion of arginine to citrulline, and this reaction is catalyzed by nitric oxide synthase family of enzymes. There are three types of nitric oxide synthase. Nitric oxide synthase 1 and 3, they are calcium dependent, and they produces a small amount of nitric oxide, which is very important for vasodilation, neurotransmission, and NOS2, which is always active and does not require calcium, it's calcium independent, it produces a high level of nitric oxide. And during chronic inflammatory diseases, NOS2 is the principal source of a high level of nitric oxide production in these diseases. So NOS2, just a little bit about NOS2. It can be induced, of course, as I said, by inflammation, cytokines, beta-catenin, TCF4 pathway, endotoxin, and also recently, David Gillers have shown that several microRNAs can regulate NOS2 expression. And also NF-kappa B hypoxia oxidative stress that can induce NOS2 and lead to a higher production of nitric oxide. So one of the interesting findings, which was reported by Stephen Ames, who is sitting here, and also Kathy Forrester, about an interaction between nitric oxide and P53. And what they found is that P53 transcriptionally represses NOS2, and nitric oxide can induce P53 activation. So they reported a negative feedback loop between nitric oxide and P53. And that really highlighted the biological evidence of nitric oxide in tumorigenesis with its interaction with the P53 tumor suppressor gene, which you know is one of the most common genetic alteration in all the tumors, most of the tumors. So I will give you some example of chronic inflammatory diseases, and one is ulcerative colitis. So ulcerative colitis is a chronic inflammatory diseases in colon, and that enhances the risk of developing colorectal cancer. This is the normal colon through endoscopy. This is the ulcerative colitis lesions. You see inflammation here. And in a small percentage of ulcerative colitis patients, after a long period of time, it can lead to the development of colon cancer. And here is the epidemiological evidence that shows that it enhances ulcerative colitis, and the risk increases, with the time of diagnosis of getting pancreatic cancer. So just briefly, because these studies are published, what was found is that there is an increased expression of NOS2 in ulcerative colitis as compared to normal colon. And it also showed a correlation with the increased serine 15 phosphorylation of P53, which is important for the stabilization of P53. And then you can see the downstream target of P53, P21 and MDM2 was also enhanced. So NOS2 expression correlated with P53 serine phosphorylation and expression of P21 and MDM2 in ulcerative colitis. So what it showed is that nitric oxide can activate the protective P53 stress response pathway and can lead to anti-carcinogenic effects, growth arrest, DNA repair, and apoptosis, and antioxidative and nitrosative stress. And the hypothesis is that nitric oxide and oxyradical associate with cancer-related mutations in chronic inflammatory disease prior to cancer, because nitric oxide can induce genetic alteration. Does it induce mutations in P53 genes before the development of cancer? So we investigated a number of different chronic inflammatory diseases, and we did find that yes, the answer is yes. There is an increased P53 mutational load before the development of cancer in these chronic inflammatory diseases, and they also showed an increased level of NOS2. So that showed an association between increased NOS2 expression, increased nitric oxide production, and p53 mutations. So nitric oxide can activate, on one hand, the protective p53 stress response pathway, as I showed here, and can also induce p53 mutation and can lead to pro-carcinogenic effects. For example, genomic instability, decreased cell cycle DNA repair, and pro-oxidative and nitrosative stress. So let me give you one example of a solid cancer, and that is pancreatic cancer. Pancreatic cancer, as you all know, is one of the most lethal cancer. It's the 4th leading cause of death due to cancer in the United States, and the median survival in advanced disease is 6 months or less. Pancreatic cancer has a huge stroma, and you see a very high degree of inflammation in that stroma. It's very active. And chronic pancreatitis, which is, as I showed in my first or second slide, increases the risk of developing pancreatic cancer. You see constitutive activation of NF-kappa B signaling, constitutive expression of COX-2 and NOS2, and as I mentioned, desmoplastic stroma, which is like more than 60% all stroma, very active stroma, and inflammatory cytokines, a number of different inflammatory cytokines. You see an increased level in pancreatic tumor. So I will give you an example of one of the inflammatory cytokines. So this is the epidemiological evidence that there is an increased risk of pancreatic cancer in patients with chronic pancreatitis, as shown first by Lowenfeld in the New England Journal of Medicine. As I mentioned, this is the H&E section of pancreatic tumors. You see huge stroma, and these are the cancer adenocarcinomas. And the stroma has a stromal cell and extracellular matrix, and these stromal cells secrete a number of different growth factors, cytokines, and chemokines that are responsible for a strong tumor-stromal interaction. And you see a stepwise accumulation from the normal to pancreatic intraepithelial neoplastic lesion, which is a precursor lesions, and then in pancreatic ductal adenocarcinoma. And what is shown is that pancreatic stellate cells is responsible for this active stroma, and pancreatic stellate cell is activated through cytokines and growth factors. And one of the very interesting observation that was published by Craig Logston is the active level of KRAS, because 90% or more than 90% of the pancreatic cancer tumors have KRAS mutations. But what he showed that not all KRAS mutant cells have the same activity. So it has to have a high and sustained pathologic level of KRAS, and that is attained through inflammatory response in the tumors. And what he showed that NF-kappa B activation of COX-2 can lead to increased prostaglandin synthesis, and that is required for the maintenance of a high pathogenic level of KRAS. So macrophage migration inhibitory factor is a cytokine, and it's a pro-inflammatory cytokine and expressed in epithelial and inflammatory cells. It regulates immune and inflammatory response. Its increased expression is found in many tumors, including the pancreatic cancer. It activates a number of different oncogenic signaling pathway, for example, NF-kappa B, AKT, ARK1 and 2, and it also enhances the expression of COX-2 and NOS2. And also, interestingly, it inhibits p53 function, and also it interferes with RBE2F pathway. And based on these and several other functions, MIF is considered as a connecting link between inflammation and cancer. So some of our work which was published by Funamizu earlier, showed that the patient who had higher level of MIF in the tumors, they had poorer survival in pancreatic cancer, as opposed to the patients who had lower expression of MIF in tumors. What he also showed that MIF expression showed increased tumor growth in nude mouse. And when you plant these tumors orthotopically in the pancreas, you see increased metastasis in MIF overexpressing tumors. So one of the important events that plays a role in invasion and metastasis is EMT pathway. And Andrew Rim showed, very interestingly, that inflammation in pancreatic cancer can induce EMT even in the preneoplastic cells. So these are the tumors. These are MIF overexpressing. These are the MIF overexpressing tumors, and you see highly undifferentiated. These are the tumors that are controlled pancreatic tumors, and they are well to moderately differentiated. And they show a change in global gene expression profile between these two tumors. And the reason I'm showing you this, because they also showed a decrease in E-cadherin, an increase in ZEB1 and vimentin, which are the markers for EMT. So one last point I would like to make, and I would finish in like 5 or 6 minutes my talk, is the microRNA and cancer. And all of you are aware very well that microRNAs play a role in tumorigenesis. And Stephen Alms, Carlo Croce have shown earlier in this report that there are many different microRNAs that are involved in several different cancers. And there are, excuse me, some microRNA that are involved in several different cancers. For example, miR-21, 190, 117, and so forth. They are subdivided into oncomirs or tumor suppressor microRNAs based on their functions, and tumor suppressor microRNAs inhibits oncogenes, and oncomirs, they inhibit tumor suppressor microRNA and leads to its degradation. Interestingly, there are several inflammatory mediators, all the cytokines, that regulates many of these microRNAs that are associated with cancer. And IL-6 is one example of cytokines that regulates miR-21, and miR-146 is regulated through Toll-like receptor and IRAK1 and TRAF6. And you see several feedback, positive feedback loop between inflammatory mediators and several different cancers. We see an enhanced expression of several different microRNA in chronic inflammatory diseases. These are the example, ulcerative colitis and chronic pancreatitis, and these microRNAs seems to play a role in these diseases. So I would like to end my talk with this slide, which shows that it's the overall balance of inflammatory signals that determines the outcome. It's a highly complex biology, and it depends on the cellular context and also tumor types. And it's the overall balance that will either be pro-tumorigenic and anti-tumorigenic. So I will stop here, and if you have any question, I would like to answer. Thank you.
Inflammatory response that mostly associated with epithelial cancer?
No. You do find in the neuroendocrine tumors too. For example, in the pancreatic PNET neuroendocrine tumors, inflammation plays a role. No, there is one. multiple sclerosis. Yes, you are right. There are some evidence that suggest a role of reactive species in multiple sclerosis. Yes. Okay, thank you very much. Stefan.
So our next speaker is Stefan Oms. He got his PhD in Germany, and subsequently he came to NCI. He's had some experience working in biotechnology companies, and he's a tenure-track investigator. Actually, he was tenured in 2010. He's in the Laboratory of Human Carcinogenesis. The title, Contribution of Oxidative Stress to Human Cancer, Evidence from Cancer Epidemiology. Stefan.
Thank you for the introduction. All right, take this one. Okay, so thanks for coming. So I will discuss more of the evidence from cancer epidemiology that oxidative stress contributes to human cancer development and progression. So there will be some overlap between me and between mine and Paris's presentation. Just not a surprise. Paris and I, we know another very well, work together and have common interests. So I will re-emphasize some of the points he already made. Probably he made it better than I am going to do. So here are the hallmarks of cancer. Here are the 6 hallmarks that were originally proposed by Hanahan and Weinberg. These are processes are needed for cancer development and progression. And then later on, there were other hallmarks proposed to be essential for cancer development. As you see, one of them is oxidative stress. The importance of oxidative stress, especially in an inflammatory environment for cancer development, is very well known and evidenced. And there's also evidence, of course, from epidemiology, and I will discuss it. What are the mechanisms leading to oxidative stress? So maybe in our context, we are looking at growth factors, diet, or altered metabolism. And altered metabolism may come from diet, but it may also come from acquired mutations. And when you have it, you get changes in mitochondrial metabolism. And then actually under, what a baseline condition, mitochondria are the major source for oxygen radicals in a cell. And then you can also have the activation of an enzyme, is called NADPH oxidase. So you get an increased level of reactive oxidant species. And what do these species do? As Paris said, they can damage the cell, but they also activate signaling pathways. One signaling pathway goes here, that leads into NF-kappa B, the NF-kappa B inducible kinase. Others, and that's a very important pathway, is actually the activation of the RAS oncogene, is very well studied. Then this pathway leads into the apoptosis signal regulating kinase 1. And then we have the mitogen activated protein kinase cascade. This is very well studied in the involvement of oxygen in activating this pathway. What these pathways have in common, they lead to the activation of either NF-kappa B and AP1, and that leads to the upregulation of pro-inflammatory enzymes. So it really goes together, as Paris already pointed out. Pro-inflammatory signaling induces the production of oxidative stress, and oxidative stress actually induces a pro-inflammatory environment. And then that's not all what we have. We have it's also observed that the expression of antioxidant, the pro-oxidant enzyme changes in cancer. So this comes from laboratory investigation, also from clinical studies. So what we see commonly in human tumors is a downregulation from enzymes that have an antioxidant activity, like manganese superoxide dismutase, that's a candidate tumor suppressor, copper zinc superoxide dismutase catalase, glutathione peroxidase. And then on the other side, we get an upregulation of pro-inflammatory enzymes. Paris mentioned them already, cyclooxygenase 2, lipoxygenases, and also nitric oxide synthase type 2 in human tumors. So overall, and we see that really in clinical specimens, this is not just in experimental systems. And we have a pro-oxidant state that is common to cancer cells. It's very well documented. And then many cancers, poorly metabolite hydrogen peroxide is also very well documented. And one might be to say hydrogen peroxide is not just a radical that causes damage. It's really a mitogen, which really induces proliferation of cancer cells. So, as I mentioned, or Paris mentioned, we are very interested in the relationship between oxidative damage and cancer, how reactive oxygen and nitrogen species actually contribute to disease onset and progression. And one can study actually this relationship with different approaches. Paris really very nicely emphasized molecular and cellular research. Nutrition research is also very important, and then animal studies, that is what Paris is doing. But you also can use observational research and intervention trial. This belongs into the domain of epidemiology. Now, epidemiology really doesn't measure directly oxidative stress, cannot. It focuses on what happens in human populations, and once make a point in human populations that something that relates to oxidative stress is related to cancer development. So epidemiology has to use surrogates, something you can measure in a human. Either markers, you can go for tumor markers, but usually blood-based markers, and there's, or just measures, has actually survey data and just assess, looking at service, looking at exposures, and uses the evidence from laboratory-based research that this exposure is actually linked to reactive oxygen production. So when I say, epidemiology, I cannot tell you we measured, the epidemiologists measured reactive oxygen species and linked it to a disease. I don't. Okay, that's a limitation of observational studies, but they are very important because epidemiological studies establish a link in a human population. So, epidemiology has looked at the relationship between oxidative stress and cancer in the context of infections and chronic inflammation, environmental factors, occupational factors that are all linked to oxidative stress and have then been linked to cancer. One can also look at diet. Now, how do diets, relate to oxidative stress? There are diets, we believe, that increase oxidative stress, and this typically is a Western-style diet. And then there are diets that may actually reduce oxidative stress, and this is a so-called healthy diet, rich in vegetables and fruits. So then comes the question, is there also oxidative damage by inherited predisposition, or are there individuals with an inherited pro-oxidant state or excessive oxygen production in their body who have an increased risk of developing an illness like cancer? What's the evidence for that? I think there's good evidence. We know some high-risk population for cancer, patients with Wilson's disease, and then hereditary hemochromatosis, a disease that Paris already mentioned. Subjects with Wilson's disease, they have a high penetrance mutation that lead to excessive copper storage. This is a copper storage overload disease. They have neurological disorders, but they have also liver degeneration and then actually an increased risk of liver cancer. And hemochromatosis patients, they suffer from cardiovascular disease, but they also tend to have liver disease and also an increase of liver cancer. And both diseases clearly have been linked to excessive reactive oxygen production in the liver. So this is diseases we have clear evidence that those kinds of predisposing mutations lead to excessive reactive oxygen production and exposure to cancer, here liver cancer. Then we assume that there are also in the population of people who are at increased risk, or at a low or moderate increased risk. And where would that come from? We think that comes actually from polymorphisms in host defense genes. in the general populations. And we know that there are genetic variations in the population that encode for proteins with altered protein function. It's really nicely shown for superoxide dismutase. And for an enzyme, it's called NADPH genome oxidoreductase. But there are also really polymorphism, well-known polymorphisms affecting promoter activity of cytokines, where they really change baseline expression of cytokines. So you have one polymorphism, you have a low expression of a cytokine, and you have another polymorphism, you tend to have a high expression of a cytokine. And that should also affect the pro-inflammatory environment. And then we have evidence from genome association studies. They identified numerous genetic susceptibility loci that predispose to ulcerative colitis. Perez pointed that disease out. It's a chronic inflammatory disease of the colon. And they point out that actually some genetic variants predispose to the disease, and many of these variants actually affect regulators of immunity and autoimmunity. So yes, there is clear evidence that there is a predisposition in the population for inflammatory diseases, and many of these inflammatory diseases have then been linked to an increased cancer risk. Okay, good. And here actually in my laboratory, we studied a polymorphism in the manganese superoxide dismutase gene. It's a Val16 alanine polymorphism. In the leader sequence of these superoxide dismutase protein, you can have an alanine or an valine. This leader sequence is important for the translocation into the mitochondria. Superoxide dismutase manganese superoxide is a mitochondrial protein. When you have the alanine, then you are more efficient in translocating into the mitochondria. So you tend to have a higher mitochondrial activity of this enzyme. And then people say, yeah, and then you should be perhaps more effective in detoxifying superoxide radicals that are actually produced in the mitochondria. So what we found is actually that carriers of the ALA alleles here, ALA, that among breast cancer patients that have poorer survival than carriers with a VAL genotype. And one would say, what does that mean? Now, when you have the alanine, I just told you, you are more likely, you're more equipped to have that a protein expressed in the mitochondria. You detoxify superoxide better. So when you detoxify superoxide better in the mitochondria, you seem to have a poorer outcome. Why? How could that be? I just said, that actually an excess of oxygen radical contributes to cancer development or progression. Well, this is actually related to what we think, and we have done a sub-analysis by treatment. It's related to treatment response. So many chemotherapeutics, when you take them, their cytotoxicity is actually also related to the production of superoxide. Actually, it's a very important part of the cytotoxicity of these drugs. We make the point when you have this genotype, you are more effective in detoxifying these superoxide radicals that are produced in chemotherapy, and you do not respond as well. So that's why you have a poor survival. So it's superoxide radicals, so may actually, or the metabolism of superoxide radicals may relate to disease development, disease progression, but also response to chemotherapy. And that can actually go in opposite directions. So when you have one genotype, it may support disease development and progression, but when it comes to therapy, it's just the opposite effect. One has to think about it. So it was a surprising result. Okay, good. Here are the cancer causes worldwide. As we see them, we think that nutrition or a bad diet contributes to about 30 to 35% of cancers in worldwide, tobacco to 15 or 30%, chronic inflammation 10 or 20%, and then the other causes are less important. So let's first look at the link between nutrition and cancer. And what do we know about the oxidative stress as a component of this link? That actually oxidative stress really links a poor diet to cancer development. All right. So how do we know that nutrition is linked to cancer? This can really come, the best evidence is really from epidemiology. So here, So here people did, very important, migration studies, where they followed people that migrated from low-risk countries into the United States or into Europe, and then observed that these populations that migrate into this country actually adapt the risk of this country very quickly. It means they have an increased cancer risk when compared to their home country where they came from. This cannot be an adaptation. This must be environmental exposure. And what we know, what we do, one of the most important things, they change that. And this has migration studies have really shown nicely how a change in a diet changes cancer risk in migrating population. Then we have the typical association studies. Typical association study would ask question, have a questionnaire, and ask you about your diet or some exposures, and then ask controls, people who do not develop a disease, and ask cases who have developed disease, and ask the question, is there a difference in exposure? I mean, if there is a different exposure, we believe that this different exposure has something to do with disease development. And there comes a lot of evidence from those studies that nutrition is linked to cancer, and then we have intervention trials. That's the last. That's the most valuable study you can have, but it's usually the last study that you will really undertake to show a link, because it's very expensive to do an intervention trial. An intervention trial would be that you have a population. You ask that population to stick to a certain diet. You ask them, or you give us a population a nutrient, a vitamin, or and then you compare it versus another group. Follow them for over years. Usually you want to do it in a high-risk population because you don't have the time to go for 10 or 20 years. And when you do it, then you ask the question, those who have the intervention you think it protects you from cancer, do they really develop less frequently the disease? And that's really what we think is a gold standard for showing that a certain dietary component is actually linked to cancer development. So, but from these three types of epidemiological studies, the common message is that certain foods, high intake from these certain foods, confer a low risk of cancer. And I just list them here. It's fruit, veggies, tea components, polyunsaturated fats, dietary fish oil, and selenium. That's a very good example. And then you have foods where the high intake of those foods confer you a high risk of cancer. That's alcohol, red meat, animal fat, salted fish, charbroiled foods, salt preserved foods, and then contaminated foods. So just focusing briefly on those diets that confer a low risk, when you really look at those diets really are typical diets that are reflective of an antioxidant activity. And that's really what people saw very early on and said, well, and they all have an antioxidant property, then protection against oxidative stress must be very important in preventing cancer development. So that's really one of the strongest evidence that we have. That actually diet is linked to cancer risk by interfering with oxidative damage. So let's look at that a little further. So here's an animal study. I told you there's nutritional research is also very important. One may say a diet that confers high risk, when you look at the upper kind of food components, is what we call typically a Western style diet. So people have now focused on Western style diet to show whether Western style diets, and they have linked an association study to cancer, to increase cancer risk, really cause disease by causing either a pro-inflammatory environment or causing oxidative stress. And you cannot do it in humans. You have to, actually some studies are even done in humans where they looked at urinary excretion of some markers of DNA of oxidative damage, and they saw that some of these markers may change with intervention, but it's really better when you do animal research. And here they fed mice a Western style diet, high in fat, low in calcium, vitamin, and they saw in mice that this diet induced pro-oxidant redox changes and inflammation in the exposed colon. So the inflammation in the exposed colon was, for example, that you have lamina propria macrophage infiltration. So macrophages go into the mucosa, and you really can see that there is inflammation in that mucosa. And then you can see systemically that you have markers, inflammatory proteins or markers for inflammation change actually with the diet. And here is a control group. Here are the animals that received the Western style diet. And you see many of the markers, they makes a point that these are all inflammatory proteins. May be true, may not be true. But most of them are up. And they say, yeah, there's a systemic inflammation among those animals who have a Western style diet. And actually, one can measure many of these same markers when you compare an obese versus a non-obese human subject. You see that the same markers actually are up in people who we would consider obese. So there is really good evidence that there's a systematic inflammation induced by Western style diet. And we would then think, and I told you many of these studies are indirect. People do not measure really oxidative stress. They cannot, but we assume that's also an underlying mechanism that then later leads to cancer. So, and then I want just to highlight a little more. So far I gave you the impression it's all clear. You know, the evidence is absolutely clear. Link diet to cancer and oxidative stress is involved, and the diet components have an antioxidant activity, actually protect against cancer in the population. We are still pretty sure that's true, but I want to point out that when you do epidemiological research, things don't work out always so easy. So people have studied this relationship between diet and dietary component and cancer development in many studies, in many very good studies. So especially in the 1970s and 1980s and still until 1990, people did some case control studies. Many of the results from some case control studies appeared in the press, and they studies always, easily said, you eat fat, you get cancer, different types. Lots of positive associations, red meat, pork, whatever. It seems like to be all clear. And afterwards, people started to do really good studies, and it's called a prospective study. Here you enroll, so case control studies have certain biases, people who have a disease. These are studies, they ask people with disease, can you remember what you had? And people with disease do actually remember exposure differently than controls. So people did prospective studies and said, okay, enroll 100,000 people, give them a questionnaire, follow up the questionnaire after 5 years, after another 5 years, and follow them up for 20 years, and then see how their exposures as a diet relates to cancer development prospectively. That's the best what you have. Large prospective studies are very expensive, take a long time, so it took quite a while to complete them. So we have data from many prospective studies, and then I just show you what came out of those. So many prospective studies looked now again at the relationship between dietary pattern and colorectal cancer. And it came out that the risk of colon cancer is increased, in fact, with patterns characterized by high intake of red meat, processed meat, what I said, a Western-style diet, and decreased with diets labeled as healthy in the meta-analysis. But I can tell you, there were significant association, but they were not very strong. Not as you would say, oh, well, you are really strong risk when you have the wrong diet. Then people always said, dietary fibers, either a high fiber diet, it protects you against a colon cancer. Actually, lots of pooled analysis of certain prospective study that did not support that hypothesis. Did not. Actually, there was even an intervention trial that failed. Then fruits and vegetable intake in breast cancer. There were some data out of the 80s and the 90s, also probably not so well-designed studies that said, yeah, fruit and vegetable intake protects you against breast cancer. Is that true? So here, meta-analysis of prospective studies suggests that high intake of fruit and vegetable combined lead to actually significant reduction, but again, it's a rather weak reduction. And then for vitamin A and C intake, folate intake, and the relationship to lung cancer, this was studied very extensively because, and I get to it later, lung cancer is really one of those cancers where reactive oxygen species are really involved in disease development and progression. And there's no question. So people thought, give smokers these antioxidants, and they are less likely to develop the disease, or look in the population, when people took these vitamins, where they protected against the disease. And what came out, no, it's a weak relationship, so it was not easy. We could not easily link antioxidants to a reduced lung cancer risk. And then here with fruits and vegetables and prostate cancer, again, there was no significant association. So I want to mention that now that I give you the wrong message. So epidemiology has not been that successful to really link individual food components that have antioxidant activity, as we think that it really protects you against cancer. So we think now it's really the overall lifestyle, really diet together with other things that protects you. And just looking at individual food components makes us not very successful. So it's maybe a flaw by study design. All right. So I told you that the kind of food we are eating is related to disease development. Poor diet increases the risk of cancer development. That's one way to look at diet, but it's not just the composition of diet, it's also the amount of diet or food that we take in that's linked to cancer. It's more how much energy do we take in and how much energy do we spend. We take in more than we spend. We gain weight, we become obese. And as you all know, obesity is really something that's a pan-epidemic here in the United States and in other developed countries. And this is a graph summarizing the development of obesity up to 2000 and shows how the prevalence of obesity have increased among men and women. And this even went up even further. So indicating that, a pretty good fraction of the population is overweight or obese. How does that relate to what we are discussing today? And that Paris actually pointed it out. Obesity is actually is a metabolic disease, but is an inflammatory disease. Obesity is linked to systemic inflammation. Here you see weight gain, and here is the extent of pro-inflammatory signaling goes just together. It's really an inflammatory disease affecting many different organs, from brain, fat tissue, liver, muscle, blood vessel, and then what Perez pointed out, the pancreas. What we have increased free fatty acids, we have decreased adiponectin, and then we have increased cytokines. These are really cytokines, the increased release of cytokines are the pro-inflammatory mediator. And what we also observe actually in most of these tissues from obese subjects, that we have infiltration of macrophages into their tissues. And that's actually seen from fat tissue to fat tissue, muscle, blood vessels, and here's the pancreas. And it's not just the number of macrophages that get into these organs, it's also the polarizations. These are M2 macrophages that are pro-inflammatory, and actually they are pro-oncogenic, you can say pro-macrophages. So, and here's the signaling we are getting. We lose adiponectin signaling, we lose omega-3 fatty acid signaling in an obese environment. We have uptake of lipids into cells. We get signaling by saturated fatty acids and by these cytokines that are in the circulation. And what we are getting again, and I showed you that before, and I made the point, when we have oxidative stress, you get activation of several signaling pathways, and they really come together at the end by activating NF-kappa B and AP1. When you look at obesity, these are the same transcription factors that are activated. So and this again leads to the secretion of pro-inflammatory molecules. And here's actually an estimate that you have an excess of 20 to 30 million macrophages per kilogram excess fat in humans. So that's an estimate. So it's a real, clearly an inflammatory disease that leads to oxidative stress, and actually to the same signaling that you have under oxidative stress. So how is that important? In 2004 came this provocative publication in Oncogene. They said overweight and obesity may account for 10 to 20% of cancer deaths in the United States, which is a big number. And they estimated, so they compared subjects with BMI 30 to 40 versus subjects with up to 25 BMI, that is normal weight. And then they said, the risk for women increases by about to die from cancer increases among women by 88%, among men by 52%. So they indicated actually that obesity may affect women more than men in terms of cancer mortality. But this is only an average. You see, that's very heterogeneous. So some cancers are very much affected by obesity, or the risk to die from this cancer really goes up among obese subjects, while other cancers, the risk doesn't go up as much. And so you have really to look at the different cancer sites. So this actually, that number, 10 to 20% of cancer deaths in the United States, that was a little controversial. That came some other reports later on that said, no, it's not that much. It's an overestimation. And in 2010, there was a paper from New England Journal of Medicine. It actually came out here from NCI. They summarized again the mortality, cancer mortality associated with body weight. It's shown here. They looked at various diseases, cancer, cardiovascular disease, and other causes associated with increasing body weight. One is a reference group. That's a group from 22 to 22.5 to 24.9. So that's average weight. And then you see, as you gain weight, increased body mass, your risk goes up. This is a relative risk. It's 5%. Oh, let's go back. 5% increase, then comes 12% increase, 34, 47, so 70. So it's a linear increase for the risk of dying from cancer with an increase in body weight. So that confirmed the original study. And this actually study also addressed another concern. People originally said, when the other ones, when they looked at the relationship between obesity and cancer, they didn't account really for smoking. And then there was an argument, smoking is actually in part responsible as a confounding factor. And in this study here was conducted among subjects who never smoked. So that confounding factor was eliminated. So that's a really strong study. So, okay, that really links obesity to increased cancer mortality. There's no question about it. And I made a good point here. Obesity is an inflammatory disease. It's chronic stress, chronic oxidative stress. So now let's go to the cancer sites that have been most closely linked to excess reactive oxygen nitrogen production. Here's a list. I took that out from here, and actually the top cancer site is lung cancer. It's interesting. Then of course liver cancer should be high, stomach cancer should be high, liver cancer, HPV, HCV, typical infections that lead to liver cancer. You have a pro-inflammatory environment. Stomach cancer, Helicobacter pylori. Bladder cancer is also some infectious disease related to it. Colorectal cancer, I will come to that. Lung cancer is number one. Why is lung cancer number one? Lung cancer is mainly caused by tobacco smoke. And there's very good evidence that actually tobacco smoke causes oxidative stress. And it's very important. It causes also a pro-inflammatory environment. Here it's a marker for oxidative damage in the oxidative damage of the DNA. Excretion of this marker into urine is increased by 30 to 50% among smokers. Then experimental studies clearly showed that many of the components of tobacco smoke reactive oxygen generating compounds, actually reactive oxygen species is produced burning a cigarette. Then we know from lung cancer, from smokers, there's a massive induction of monocyte recruitment and activation in the lung tissue. It's clear. And then we also know that cytochrome P450, so when you smoke, you inhale a lot of carcinogen compounds that are then further metabolized by P450s. And actually, when you have this metabolism, P450-mediated metabolism, a byproduct of it is also oxygen radicals. So altogether, really generates an excess of oxidative radicals and oxidative stress in the lung of smokers. Then we know that asbestos exposure or silicosis is associated with the development of about 4,000 to 10,000 cases, lung cancer cases in the United States. And this is absolutely clear. This leads to persistent inflammation when you get asbestos or these silicon little crystals into your lung. that can be measured clearly. So you have a generation of radicals at the particle surface and particle activated cells. This is really an inflammatory disease, and that leads to lung cancer and asbestos mesothelioma. Radon is another important source for lung cancer development. Radon also produces oxyradicals. It's pretty well known. And then we have also the association of asthma and chronic bronchitis with lung cancer risk. When you are an asthma patient or you have bronchitis chronically, you are at an increased risk of developing lung cancer independent of smoking status. So all over that clearly says, lung cancer, the major causes of lung cancer all produce excessive oxygen radicals. So because of that really kind of convincing evidence that this is a disease that is linked to reactive oxygen species, people said, okay, let's give a subject at high risk of developing lung cancer nutritions or vitamins from which we think they are antioxidants. And then ask the question, we have one arm of smokers who get these supplements, and we have another arm of smokers who don't get these supplements, and follow them up. And that was a large prevention trials you see. And we would expect that those ones who have the vitamins, the protective vitamins, develop less lung cancer. People did it, and what came out was actually rather disappointing, very unexpected. There was no difference, or when you took the antioxidants, you were more likely to develop lung cancer. People were really, how could that be? They took actually several years of discussions and additional research, and what people found is that pharmaceutical doses of antioxidants that are apparently non-toxic bioactive components, antioxidants, in the general population might have adverse effects in individuals with high-risk behavior, such as high smoking, alcohol intake. They actually said they behave like pro-oxidants in this population, and they showed it with experimental data. So essentially, the trial failed by design. And that's also how we have to think about many of these antioxidants, that they are, in certain situations, in certain doses, they do exactly as you think. But in other situations, and in a higher dose, they may just do the opposite. All right. Okay, good. And then what is another cancer where we have really good evidence for the involvement of reactive oxygen species, and that's colorectal cancer. And in colorectal cancer, I think that's really the cancer where epidemiology made the biggest impact. And what was that impact? People looked at non-steroidal anti-inflammatory drugs and asked the question, how is the intake of these drugs related to cancer risk? And for prostate cancer, for colon cancer, the epidemiology was as clear as it could be. I think almost as clear as tobacco smoking causes lung cancer. You rarely have it so nice in epidemiology. And there was one study that showed, that was actually one of the early studies that these non-steroid and anti-inflammatory drugs increase colon cancer risk. But all other studies, so many, really came to the conclusion, you're a regular user of non-steroidal anti-inflammatory drugs, your colon cancer risk goes down by 50%. It's absolutely clear. And that's, and what do they do? They inhibit inflammation. Said, well, inflammation must be very important in colon cancer, and that is a human disease. So really made a very big impact. And I just show you here again that the same graph that Paris showed you, that actually regular use of aspirin reduces the risk of cancer deaths in general. Those are the people, it's a control group. This is a group that took aspirin. And it cuts really the risk of cancer. It's really best for colon cancer. The association of aspirin with any other cancer is weaker, I have to say. For colon cancer, it's absolutely clear. So there are people who say there are few things people should take really to protect themselves. And a tablet of aspirin every day, if you have not the problems with bleeding, is one of them you should take. Okay, good. And then let's go to chronic infection. We said it's 10 to 20% of all cancers in this world are explained by chronic infections. Viruses cause about 10 to 15%, bacteria about 5%, and I will discuss you this point. Here's an infection associated with human cancers. It's listed. Paris showed you already some of these data. This evidence is clear. So there are certain viruses are certain are related to the to certain cancers, hepatitis B and C, to liver cancer, HPV, to cervical cancers, and so forth. And then here Helicobacter pylori to gastric cancer. And these are actually two parasites that are related to bladder cancer and cholangiocarcinoma develop in certain parts of this group. And this is not something we propose, that's proven. So that's a really nice data. Now, in terms, I gave you that number, bacteria account for about, bacterial infection account for about 5% of all human cancers. I think that's something that's floating at this point of time, because people start now looking at the human microbiome and disease, and they get actually, new insights, things that they have not accounted for. Let's briefly go into that research of human microbiome and disease, especially cancer development. First of all, we are colonized by a whole lot of bacteria. This just shows our genome versus the genome of the bacteria. The colonizer, the huge difference. And our skin is just full with them, gut, lung, oral cavity. And most of these bacteria, we don't know actually what's that. We don't know species. So we learn more about them. And what people know at this time is that a bacterial dysbiosis is associated with disease, several different diseases, but also cancer. What is a dysbiosis? It's a loss of commensal and gain of pathogenic microbes. So we have many of these microbes. We say, yeah, there's symbiosis. It's good that we have them. And then there's also pathogenic ones you don't want to have, or you don't want to have many of them. And they say in disease that shifts actually towards the pathogenic ones. Measured. And then there's general loss of diversity is a characteristic of the disease state. And here I took that from a Nature review that shows the microbiome of healthy volunteer. These are healthy volunteer in this study for inflammatory bowel disease. And they show here how that shifts actually in patients. So you get a different microbiome in the patient population. Type 2 diabetes, you see the same. This must be all gut flora. And then here is enterocolitis, necrotizing enterocolitis. That is a disease that affects premature infants. And that's a disease that actually is an inflammation of the intestine and the colon. People don't know exactly how it works, but because they think it has something to do with the microbiome, and that's why they studied it, and they showed that in infants that are affected by it, it's completely different. So people are working on it, try to understand it, because so far we cannot do much when infants present with that kind of a condition. Okay, but the take-home message is, in disease stage, the microbiome is changed. And then comes the question, is it just changed, or does it change the disease? So is it pathogenic? All right. So here came a summary in New England Journal of Medicine. And I took it. I don't show you the paper. They summarize the finding of a paper. And that paper reports that bacterial translocation is a driver of disease progression in colon cancer. So these are not bacteria that we had linked before to cancer. Said, oh yeah, this is a cancer-causing bacteria. No, these are just, we have the gastrointestinal tract. You acquire genetic mutations, and while you acquire these mutations, it leads to loss of junctional proteins and diminished production of mucus in the colon mucosa, in the epithelium, intestinal epithelium. And what happens is bacteria can now translocate, and they can translocate abdominal lymph nodes. And when they do it, they actually interact with myeloid cells. That's was shown, the secretion of interleukin 23 that activates T cells. They secrete interleukin 17. And that interleukin 17 now activates STAT3. And STAT3 is one of the, a key oncogenic gene in human carcinogenesis. Activation leads to proliferation. So they said actually that this translocation starts driving carcinogenesis. It induces proliferation. So it's not just that there is perhaps a change. No, that the change becomes a drive. And then here are several other papers. Yeah, now actually many papers, and I just encourage you. I give you here just some examples here. a paper published in JCI, dysbiosis predisposes mice to transmissible colitis and colitis-associated colorectal cancer, meaning there's a dysbiosis in one animal that can actually infect another animal. That's what this study is saying. And that study also identified one gene that is perhaps very important in regulating actually the composition of our microbiome. Then, other study, obesity-induced gut microbial metabolite, deoxycholate. That's produced by gut bacteria. It gets into the bloodstream, gets to the liver, first pass effect, and promotes liver cancer by inducing a senescence-associated secretory phenotype in hepatic stellate cells, leading to release of pro-inflammatory factors. Actually, the pro-inflammatory factors at the end drive liver cancer. But it's really interesting. It's actually a metabolite from bacteria in the gut that initiates all of that. And I can tell you, this metabolite, we have the metabolome of breast tumors. We find it accumulating in breast tumors. We have those data. We're actually interested in the metabolite. It's not just that it goes to the liver. It goes actually also to other organs. And we are interested in wanted to know what does it do in breast cancer. Then hepatocarcinogenesis in chronically injured liver cells depends on the intestinal microbiome and toll-like receptor 4 activation, and they show actually LPS released by bacteria activates this pathway and that promotes disease progression. Again, here, bacteria, the microbiome becomes causal for, mostly here actually for disease progression. So people have not looked at that before. So now people start thinking, what can we do? How can we treat the microbiome of cancer patients and perhaps in slowing down progression? So it's important new information. Now here, last slide really relates to the microbiome. So in 2012, there came a paper out in Genome Research that reported that a certain family of bacteria, the Fusobacterium, they accumulate in some patients with colon cancer. Let's see at the peak. This patient has a very high colonization, this patient, this patient, this patient. So it's heterogeneous. And then again, how could it happen? Is it of any consequence? Independently, other studies showed, and we think it is of a consequence, that exactly this family of bacteria activates a Wnt signaling pathway. And the Wnt signaling pathway is very important in colon cancer progression. So we think, when you have it, it should actually promote colon cancer progression in humans. So it's bad, so you want to get rid of this bacteria. Then there are some other, this is also, look just in these papers I write down here, and then there are some other papers that describe now bacteria that colonize the gut, and some of them, they are not so well described. It's all new research. They think some of these bacteria have genotoxic effects in the epithelium, and some bacteria cause actual reactive oxygen species production in the epithelium. Again, driving disease. So that's something. So we really don't know to what extent bacteria contribute to cancer development and progression, but with those data, it appears that at least colon and liver cancer could be much more affected by bacteria than we thought, and perhaps distant tumor sites too, and that has to be explored. So good, now let's go to the last cancer. Any individual cancer cause less than 5%. High penetrance gene, I will not highlight. So certainly skin cancer is a disease, as we believe, is caused in part by reactive oxygen species. Skin cancer is a very common disease. I give you just here the incidence for 3 categories. Of those, melanoma is the most important because it's relatively deadly, and there are 76,000 cases per year. And then what causes skin cancer? We know sunlight. We know that UVB radiation causes signature mutations in the skin. But then we know also that UVA radiation generates oxyradicals in the skin, and believe that these oxyradicals actually are important for disease development. With humans, again, we would have to, you do something like a cream on your skin that inhibits reactive oxygen species, and we show that that would inhibit the risk of skin cancer development. I think we don't have those data, but we have good data from experimental systems that really shows that UV radiation promotes skin cancer because it causes oxidative stress. One thing we know is that UV radiation reduces SOD catalase in skin, and we know that all skin promoters, so that agent, you put them on skin, they promote skin cancer development. These agents, all of them, produce excessive oxygen radicals. And then people work with those promoters, they change it chemically a little bit, and the ones that become inactive, they don't produce oxygen radicals. It's pretty well shown. So from that, people really think that reactive oxygen species in skin cancer development is very, very important. And then here, ROS, reactive oxygen species in cancer progression, people really think that the activation of the NF-kappa B pathway is a key pathway. And I mentioned that several times before. And in terms of environmental exposures, what do we know about activators of the NF-kappa B pathway? Oh yeah, we know a lot. That environmental hazards, infection, physiological stress, and inflammation all cause the activation of this pathway. All right, so here's a summary. There is strong evidence that oxidative stress is involved in human cancer causation and promotes disease progression. I hope I convinced you. And Perez, of course, is a lecture. I'm just add on to it what he already showed you. Cancer epidemiology has not succeeded in giving specific antioxidants to a reduced cancer burden. I have to say that. Still, I think cancer epidemiology shows that that reactive oxygen species should be very important. And then knowledge of tumor redox biology promises better prevention strategies and new treatment options. Thank you. Any questions? Please.
But the food rich antioxidants are for cardiovascular or reduction of the enzymes or antioxidants.
Okay, good. So I think you make a very good point. So, the point with antioxidants, I think also from the clinical trial, is they are beneficial in a certain context. And people think, in the overall lifestyle, overall diet, in that context, antioxidants are very good. But when we try to mimic it and just giving certain antioxidants, then we don't find the effect. And the approach may be wrong, so people think so. It's approach, maybe the dose is too high. It does not invalidate the assumptions that fruit and vegetable intake is beneficial. I think that's the take-home message at this point of time. Also, people are still working and trying to figure out, how should you deal with antioxidants? Many people still take them. You buy them in the store. People take lots of them. I think one would say now there's no evidence that it's good. Actually, there's no evidence it may be bad and you take a lot of them. In terms of upregulation of antioxidant enzymes, so this is also a very difficult kind of a question. So people say that good data is for tumor growth, the primary cancer cell really needs excessive oxyradicals because they are mitogens and promote the growth of the cancer. They say about perhaps there's a switch when you become metastatic. So in the metastatic situation, there's actually also a metabolic switch. And that metabolic switch is away from macromolecule synthesis that supports growth towards protection against oxyradicals. So in the importance of the expression of an antioxidant enzyme may be different at certain stages of disease. And people know that also from the manganese superoxide. The people are all confused. They say, in my model at that stage is up, in the other said that my model at that stage is down. How can it have the same effect, And the answer probably is, in certain contexts, the cancer cell suppresses it because now it's good. Now it wants a reactive oxygen species. In another context, it cannot afford it and then it regulates it up. But there's not a global answer to it. But I think there is agreement, you know, with metastatic cells and a switch in metabolism towards more protection, less of the growth, survival now. That is probably true. So I cannot give you another answer that, oh yeah, no, high expression of superoxide disputases protects you against cancer. It seems not to be. So, oh, okay. No, it doesn't. I think they are extremely good, you know, for certain pathways. There are, in cancer, it's absolutely clear from experimental models and also how we then look at the disease that in RAS mutant cells, they really depend on superoxide production for growth and survival, and actually very nice experimental data when you block mitochondrial metabolism that, or you block superoxide radical formation, or you block the pathway that is actually activated by the superoxide radicals, then you block the growth of these cancer cells. So they are dependent. So, I mean, you know, how do you use that information? I think you have to interfere at you know, it's against this precision medicine. You have to interfere at a certain stage where it's of benefit. You may say it's the same therapy would also work at as another stage, and it may not. It may not, yeah. So you have to have all the information. Okay. Right. All right. Thank you.
Short and sweet.
Okay, yeah.